Lectures. We're going to talk about the ear physiology. So last time we looked at all the ear structures and we talked about how they're going to play different roles. So some of them are going to play a big role in our hearing. Some of them are going to play a bigger role in kind of our equilibrium. And now we want to just look at each one of those little structures and different kind of parts of that internal ear and see what their jobs are. Okay, so here is our sound summary. Okay, and we've talked about this a little bit already. Okay, so sounds set up vibrations in the air that beat against the eardrum. So everything again is coming in to that ear. It's going to run up against that eardrum. This is going to push a tiny chain of bones that press fluid against our internal membranes. Forces produced by the movement of fluid are going to excite the hair cells in our cochlear duct, and then this is going to stimulate neurons that are going to create impulses, and then those impulses are going to kind of be sent up to the brain for interpretation. So again, it's sound coming in, hits the tympanic membrane, moves those little bones, pushes that fluid through our cochlear duct, and we're going to stimulate these hairs, and then those impulses are going to be created, and then those are going to go up to the brain again to tell us kind of what we're hearing. Okay, so this is a good picture just to kind of summarize everything. So we can see the sound waves coming in, they hit the tympanic membrane, cause these bones to vibrate, push on our window here, and then again it's going to create this movement of fluid through our cochlea, because again the cochlea is the one that's going to do our hearing for us. And then if it's the proper frequencies, we're going to stimulate those hair cells in the cochlear duct, and then we're going to hear sounds. Okay, and then here again is just our little picture of, again, we want to remember that the cochlea is the part that is doing our hearing for us. And we can see in this one, we see our oval window. Remember, these are filled up with fluids, and they're kind of creating these vibrations and this movement of the fluid. And then again, it can cross through this little cochlear duct here and stimulate those hair cells. And this gives you just a nice little summary of kind of what we just talked about. Okay, so sound waves vibrate the tympanic membrane. My auditory ossicles, or bones, are going to vibrate. Pressure is going to be amplified. We're going to create these pressure waves, okay, by the stapes pushing on the oval window. It's going to move fluid through that scale of vestibuli. And then here is the trickier part. Sound waves with frequencies below hearing travel through the helicotrema, so that's where it kind of twists and turns. We saw that in the picture here, okay, helicotrema. And do not excite hair cells. Sounds in the hearing range go through the cochlear duct, vibrate the basilar membrane, and then they're going to cause this movement or this bending of these hairs inside the cochlear duct, okay? And that's going to be what's going to create those impulses for us. And again, we saw this picture last time too, but we can see our little channels here, and we can see our little nerve kind of hooked up to those little hair cells. So this will be the cochlear duct, and again, if it is the proper frequency, it's going to move that fluid through the cochlear duct, and then it's going to bend those little hairs and again create those little impulses for us. So now we get to the other part that most people are maybe not so aware of, and this is our equilibrium. And most people, when they hear equilibrium, they think of our balance. But this is going to be the other parts of the inner ear. Okay, so the equilibrium receptors in the semicircular canals and the vestibule, because remember, cochlea is all hearing. My semicircular canals and my vestibule are going to be the ones that are going to help us with our equilibrium. But these collectively are going to be called the vestibular apparatus. They're going to initiate reflexes needed to change position or perform complicated movements, and they're going to be divided into two parts. So we have our vestibule receptors, and we have our semicircular canal receptors. And again, we want to just remember sort of which type of receptor is in each one of these and which sort of forces is it responding to, okay? Because again, those are going to be where the multiple choice questions are going to kind of come from. So my vestibule receptors are going to monitor linear acceleration and head position in respect to gravity. Sometimes they're termed as static equilibrium because gravity is always kind of pushing on us. Our semicircular canal receptors are going to monitor changes, but again, this is going to be with head rotation, and this one is going to be a little different because it's going to be termed as dynamic equilibrium. So vestibule, okay, linear acceleration, head position with respect to gravity. Semicircular canals are going to be head rotation, and again, you have that circular part, which kind of makes it easy to remember that these ones are going to help us with our rotation. So my vestibular receptors are suspended in the vestibular perilymph. There are going to be these two membranous labyrinth sacs, okay, and we're going to have a saccule. It's going to extend anteriorly into the cochlea, and we're going to have a utricle, and it's going to extend into the semicircular canals, and we'll see these in a picture here too. Okay, these are going to house our equilibrium receptors, and they're going to be called our maculas. Okay, so you want to remember that the vestibular receptors are going to be called that macula, okay, and then the semicircular canals are going to have their own. Okay, so here's our picture again, and we want to come back to this one, and we just want to remember that this middle part here is the vestibule, and this is where they've kind of broken it up into the part that leads into the semicircular canals and the part that leads into the cochlea. And you can see in the picture here that I have these two macula, and they're in each of these different little regions in that vestibule. Okay, and again, those are monitoring acceleration, rotation, head position as we get into this little part here. Okay, so here is our macula. They're going to be the sensory receptor organs that monitor the position of the head in space. Therefore, they're going to play a key role in helping us to control our posture. And again, the macula are going to be the ones that respond to linear acceleration, okay, so changes in straight line speed direction. But again, these ones are not going to be the rotation. The ones that are going to help us with rotation are going to be the ones that are in the semicircular canals. Okay, so here's our little pictures of our macula in the utricle and our macula in the saccule. And again, linear acceleration for these ones, okay? So anatomy of a macula, and again, everything with the ears kind of comes back to these little hair cells, but each macula is a flat epithelial patch containing hair cells. The hair cells are embedded in an otolith membrane. We're going to see that in a second here too. The otolith membrane is a jelly-like mass, and it's studded with tiny stones or otoliths. Okay, and you'll hear people talk about ear stones, and with especially when they get dizziness or they get any sort of trouble with their equilibrium, they'll talk about these stones. But this is more commonly known as these little ear stones. Okay, so here is a little picture here of our macula. And again, we can see all these little hairs here, and we can see this little membrane, and we can see how the membrane has a bunch of little stones on here. And what's going to happen is, as I move in different directions, this little membrane with the stones on there is going to shift back and forth. And I'm going to have a macule that's orientated up and down. I'm going to have one that's orientated side to side, and it's going to cause these bending of these hairs when I move in certain directions. And then those are the impulses that are going to be created and kind of sent to my brain to sort of tell me what my body is doing. Okay, so my utricle macula is going to be orientated in a horizontal orientation. So it's resulting in hairs that are vertical. This one's going to help me with my responding to acceleration, okay? And my saccule macula is going to have a vertical orientation, and this is going to result in hairs that are going to be horizontal, and these ones are going to respond best to our vertical movement. So I'm just basically orientating these in different ways so that I can monitor this acceleration in a straight line, so up and down and side to side. But again, these ones are not rotation, okay? That's the most important thing to remember for the multiple choice questions. So the hair cells are going to synapse with fibers of the vestibular nerve, which we learned about before, and it's going to coil around their bases, okay? And then the way we activate these is, again, starting and stopping movements in the linear direction are going to cause the otolith membrane to slide backward or forward, and this is going to cause the bending of the hairs. The hair cells are going to release neurotransmitters, and they're going to continuously send impulses up to the brain. The amount of neurotransmitter released is controlled by the amount of bending. So we come to this picture, and the cool thing with this picture is that, we're always releasing neurotransmitters, okay? So even when my head is straight, we're always releasing neurotransmitters. So it's not that they kind of turn on or off. It's just depending on the movement of my head, I'm going to release more or I'm going to release less, and it's going to be the amount of these neurotransmitters that's being released that's going to tell my brain sort of what my head is doing at this particular time, okay? But we have our macula, right? And it's going to be in the vestibule. And again, this is one that's helping me with my linear acceleration, okay? So now we get to the cristae ampullaris, and again, with these ones, we're looking at rotation. So we had the macula for the vestibule, and then when we get into the semicircular canals, what we're looking at are these cristae ampullaris, okay? So they're going to be the receptor for rotational acceleration. It's going to be a small elevation in each of my semicircular canals. Because the semicircular canals are located in all three planes of space, and again, we're going to have one in each ear, all rotational movements of the head are going to disturb at least one of these cristae. Okay, so again, macula, linear acceleration in the vestibule, cristae ampullaris, semicircular canals, and it's dealing with head rotation. Okay, so here we get our semicircular canals, and I can see my cristae ampullaris, and again, I have a left side and a right side, so I'm going to get a lot of information sent up to the brain. You can also see the vestibule, right? And I can see my macula, and I know those are for the linear acceleration. Okay, so anatomy of a cristae ampullaris. Each cristae, again, is going to be composed of hair cells, very similar to that of the cochlea and the macula. You're going to have the dendrites of the vestibular nerve that are going to encircle the bases of the hairs. Okay, so we're looking at bending hairs and stimulating these nerves. The only difference in this case is that the hairs are going to be met by rotational movements. Okay, and here's just a real picture of the crista ampullaris, which is really, really cool to kind of see up close on the microscope. So activating receptors of cristae ampullaris, so they're going to respond to changes in the velocity of rotational movements of the head. Again, rotation, our movement causes movement of the endolymph in the semicircular canals, thereby bending the hairs and sending those impulses up to the brain. Here's our little picture of somebody kind of rotating. And again, we want to kind of, it's a good kind of visual, but again, we're just trying to memorize which part of those internal structures. Cochlea is going to be hearing, the vestibule is going to be linear acceleration, and it's going to be the macula, and then my semicircular canals are going to be my rotational movements, and again, this is going to be the cristae ampullaris. Okay, and information from equilibrium receptors is going to go directly to reflex centers in the brain. And again, we do this because we're trying to correct really, really quickly, and we're trying to make sure that we don't fall down. So if we had to kind of go and take a really, really long time to kind of analyze all these things and it wasn't going to that reflex center, a lot of times our corrections would be way too late. Okay, so the last thing we're going to look at in this part of the chapter is going to be motion sickness, because this is something that everybody's kind of dealt with when they're out on boats, or you're flying, or some people even get it when they're kind of sitting in the car, you know. But this is kind of what happens. So motion sickness is when we get this little sensory input mismatch, right? So we're getting a lot of information from our ears, we're getting a lot of information from our vision, and what this does is that the input from those two structures doesn't kind of match, okay? So my visual input is suggesting to me, or to my brain, that you are in a fixed position or you're not moving, but the wave movements are going to stimulate my vestibular apparatus, and they're going to tell me that I am moving, okay? So my vestibular and my visual info are going to disagree, and this is what's going to kind of make me feel kind of sick, okay? And what we do when we take medications for motion sickness, and I always tell people, you know, if you're not sure if you get motion sickness, you're always better off kind of taking these medications, because it can really kind of just ruin your whole day if you're out on a trip. But the medications that we take are actually going to depress that vestibular input kind of from the ears, and we're going to be able to sort of just rely on that visual input.